By the end of this video, we will be able to differentiate between mitosis and meiosis. Before going into details, kindly subscribe our channel so you don't miss these interesting factual videos. Mitosis. Mitosis is a fundamental process in which a single cell divides into two identical daughter cells. This division is crucial for the growth, development, and maintenance of multicellular organisms. Here's a more detailed look at the stages of mitosis. Interphase. Before mitosis begins, the cell undergoes a period of growth and DNA replication known as interphase. During interphase, the cell prepares for division by duplicating its DNA and organelles. Prophase. Chromatin, which consists of DNA and associated proteins, condenses into visible chromosomes. The nuclear envelope begins to break down, and spindle fibers form, extending from structures called centrosomes located at opposite ends of the cell. Metaphase. The condensed chromosomes line up along the metaphase plate, an imaginary plane equidistant between the two centrosomes. Spindle fibers attach to the centromeres of each chromosome, aligning them for separation. Anaphase. The paired sister chromatids of each chromosome separate and are pulled toward opposite ends of the cell by the shortening spindle fibers. Once separated, each chromatid is considered a full-fledged chromosome. Telophase. Chromosomes arrive at the opposite poles of the cell, and the spindle fibers disassemble. Nuclear envelopes form around each set of chromosomes, effectively creating two nuclei within the cell. Chromosomes begin to decondense back into chromatin. Cytokinesis. While mitosis technically refers to the division of the nucleus, cytokinesis is the process of dividing the cytoplasm to produce two distinct daughter cells. In animal cells, a contractile ring composed of actin filaments forms around the center of the cell, pinching it into two separate cells. In plant cells, a new cell wall, called the cell plate, forms between the daughter nuclei, eventually separating them. Mitosis results in the production of two daughter cells, each with the same genetic material as the parent cell. This process ensures that each daughter cell receives a complete set of chromosomes and is essential for growth, repair, and maintenance of tissues in multicellular organisms. Meiosis. Meiosis is a specialized form of cell division that occurs in germ cells, which are responsible for producing gametes, sperm and egg cells, in sexually reproducing organisms. Unlike mitosis, which results in the production of identical daughter cells, meiosis involves two rounds of division, resulting in four daughter cells with half the number of chromosomes as the parent cell. Here's a detailed breakdown of meiosis. Interphase. As in mitosis, the cell undergoes a period of growth and DNA replication during interphase, resulting in the duplication of chromosomes. Meiosis 1. Prophase 1. Chromosomes condense, and homologous chromosomes pair up, a process called synapsis. During synapsis, genetic recombination occurs between non-sister chromatids of homologous chromosomes, resulting in the exchange of genetic material, crossing over. Metaphase 1. Homologous chromosome pairs align along the metaphase plate, with one chromosome from each pair facing opposite poles of the cell. Spindle fibers attach to the centromeres of each homologous chromosome. Anaphase 1. Homologous chromosomes are pulled toward opposite poles of the cell, separating and reducing the chromosome number by half. Telophase 1. Chromosomes arrive at the opposite poles of the cell, and cytokinesis occurs, resulting in the formation of two daughter cells, each with half the number of chromosomes as the parent cell. Each chromosome still consists of two sister chromatids. Meiosis 2. Prophase 2. Nuclear envelopes dissolve, and spindle fibers begin to form in each of the two daughter cells. Metaphase 2. Chromosomes align along the metaphase plate in both daughter cells, with spindle fibers attaching to the centromeres of each chromosome. Anaphase 2. Sister chromatids are separated and pulled toward opposite poles of the cell. Telophase 2. Chromosomes arrive at the opposite poles of the cell, nuclear envelopes form around the separated chromosomes, and cytokinesis occurs. This results in the formation of four haploid daughter cells, each with a unique combination of genetic material due to crossing over during prophase 1. Meiosis is essential for sexual reproduction because it reduces the chromosome number by half and introduces genetic diversity through crossing over and the random assortment of chromosomes during metaphase 1. The resulting gametes, sperm and egg cells, each contain a unique combination of genetic material, which is crucial for the genetic variation observed in offspring. Thanks for watching. To learn more, just visit and subscribe our YouTube channel. Link in description.